Welcome to the biology class for class 12th science. Dear students, in part 1 we have already seen double fertilization and triple fusion. Today I am going to continue with it. So let us begin. Today the topic is development of endosperm. Before that I would like to revise. This is the structure of a flower. What happens after the fertilization? So the heading I have given is post-fertilization events. After the fertilization, yesterday, that is in the last class, endosperm was formed and zygote was formed. What happens after fertilization? So dear students, as soon as the fertilization is over, petals will fall off, androecium will also fall off, stigma and style of the gynoecium also will fall off. What will remain is only the structure ovary and ovule. So we are going to discuss about the endosperm and development of zygote. Let us come to the development of endosperm. The triploid primary endosperm nucleus forms the endosperm for the nourishment of the embryo. Endosperm contains reserve food to nourish the growing embryo. What is perisperm? The endosperm receives food supply from nucellus, which ultimately perishes and remains in the form of a thin layer outside the endosperm, and this is called perisperm. Dear students, in angiosperms, endosperm develops in 3 different ways. This is an important question. In the board exam, it has been asked many a times. So here there are 3 types of endosperm: nuclear type endosperm, cellular type, and halobial type. Excuse me. In nuclear type, this is an embryo sac. As you can see, this is micropyle end, and inside this is embryo. This endosperm, it divides into many nucleus. So multinucleate endosperm is formed. Here it does not undergo cytokinesis, that is, it does not form cell wall. So these nuclei are naked and they are arranged at the peripheral surface of embryo sac. The best example of nuclear type endosperm is coconut water, which is having the free nuclear endosperm. And generally most of the plants show this type of nuclear type endosperm. Examples are maize, wheat, rice, sunflower, coconut, etc. The second is cellular type. Here this is the embryo sac, this is the embryo, and the endosperm divides into so many nucleus, and here after every division nucleus develop cell wall, that is, it undergoes cytokinesis, and that is why it is called cellular type, because it represents so many cells here, and here this is seen in peperomia plants. The third is halobial type. This endosperm development is intermediate, that is, it is having both nuclear type and cellular type of nucleus. This is the embryo sac. The nucleus divides in the starting, and half of the chamber, which is called micropyle chamber, there the nucleus, they are free, they do not develop cell wall, while towards the chelical chamber the nucleus undergoes cytokinesis, and so it is cellular type. Now let us move to the second topic, development of zygote into embryo. This particular process is called embryogeny. This we are going to study in dicot seed as well as monocot seed. After the endosperm, this zygote here you can see is single cell. It elongates, and as it elongates, it divides into two unequal cells. One is large called basal cell and other one terminal cell, and this division is transverse. This basal larger cells, they are called suspensor cell, which are towards micropyle end, while the embryonal cell are towards the antipodal side. Now these suspensor cells, they divide into 7 to 8 cells, and this division is transverse division. At the same time, embryonal cell also undergoes division, and from 1 they become 2, 2 to 4, and so here this is quadrant stage. In the suspensor cells, the upper one enlarges and become haustorial cell. This is towards the micropyle end. The function of haustorial cell, which is a very big other than other suspensor cell, is to absorb food for the developing embryo. The last cell in the suspensor cell near the embryonal mass is called hypophysis. This gives rise to radicle and the root cap in future. Now this is an octant stage. Now let us move towards the embryonal cell. The embryonal cell divides thrice and they become 8 cells, and that's why the stage is called octant stage. Now in this octant stage, the cells are arranged in tiers. The 2 tiers are shown here. The upper one near the suspensor cell is called hypobasal, and the bottom one is called epibasal. These epibasal cells will develop in future to cotyledons as well as plumule. These embryonal cells, they go on dividing and changes into a heart-shaped structure, which is nothing in the future, but they are going to be called as cotyledons. So here these are haustorium, these are suspensor cells, and these are 2 cotyledons. Between the 2 cotyledons, the notch where you can see here, plumule will develop. At the base of suspensor cell, radicle will develop. And this is how the dicot embryo is formed inside the ovule. Now let us move to the monocot embryogeny in the monocot seed. Here also the zygote is single cell. It will undergo transverse division just like monocot and dicot have both the starting divisions are similar. So basal cell is formed on the top towards the micropyle end, and terminal cells, they develop into embryonal cells. As you know, the suspensor cells on the top becomes larger and is called haustorium, which is, the function is to absorb the nutrition for the development of the embryo. This is quadrant, as you can see. In monocot seed, you know that they have single cotyledon, which is later going to be called as scutellum also. So it is similar. Radicle is on the bottom of the suspensor cells, which is called hypocotyle, and these are the middle cells of it. Now, later on it develops into this structure, in which cotyledon is called scutellum. Here you know that the coleoptile is a sheath-like structure under which shoot germinate, that is plumule, and at the bottom there is a sheath-like structure called coleorhiza under which radicle will develop. So these were the development of embryo in dicot seed and monocot seed. On the base of the endosperm, there are 2 types of seeds, non-endospermic seeds and endospermic seeds. What are non-endospermic seeds? Endosperm is used up in the development of the embryo. Such seeds are called non-endospermic, and in them food is stored in the cotyledons. Examples are gram seeds, pea seeds, bean seeds, pulses, etc. Endospermic seeds, here the endosperm persists after the development of embryo, and here cotyledons are definitely small. So examples are rice, maize, cereals, etc. So dear students, hopefully you might have understood. These are important questions. These three endosperms are asked in exam. This full development is also asked, as well as monocot development is also asked. So today we have completed second chapter. Thank you and have a nice day.